Assalamualaikum warahmatullahi wabarakatuh. Greetings, lecturers and friends. We are from the second BioCell Group project, and today we will be talking about the roles of lower eukaryotic cells in pharmacy.
Okay, so we recall that eukaryotic cells are those that contains membrane-bound organelles. This time, however, we will dive deeper and learn that there are two types of eukaryotic cells, which are the upper eukaryotes, such as the plant and animal cells, and the lower eukaryotes, such as the fungi and algae. Today, I wanted to highlight the two very significant roles of eukaryotic cells, and especially the lower cells, which are the fact that they are host to many recombinant proteins and are able of synthesizing secondary metabolites. But why are these important? Well, in the case where the human body loses or experiences decreasing ability to produce these proteins and necessary metabolites, fungi can help synthesize these proteins through a lengthy in vitro processes. The products of this process are later used to produce the supplements, drugs, and other pharmaceutical products that we can consume in order to fulfill our body's needs.
Okay, so now I'll be talking about the advantages and disadvantages of lower eukaryotic cells. One of its advantages is its ability to produce authentic human eukaryotic proteins. We all know that prokaryotic cells can produce proteins, but it can only produce simple ones, whereas a human protein is really complicated, so we use lower eukaryotic cells instead. Another advantage is it is a production host to many biopharmaceuticals. It is because we can actually manipulate its genes and cellular pathways to produce these proteins. And there are other advantages, such as it has a high growth rate and it can be grown in small vessels or large bioreactors. However, it also has disadvantages such as limited post-translational modifications possible. This is because the strain that we have to use should be modified first for it to produce the proteins, and also incorrect protein folding, which leads to low yield of non-fungal proteins. If there's incorrect folding, it means that the cell produced will be inactive and it will die, so there's no point in using. As mentioned in the previous slide, one of the role of a lower eukaryotic cell is synthesis of secondary metabolites. We know that lower eukaryotic cells, such as fungi, can produce metabolites which are classified into primary and secondary. Primary metabolites are used by the eukaryotic cell for growth, whereas secondary metabolites are secreted as protection against pathogens. In pharmacy, we use the secondary metabolites to produce drugs.
Okay, so we will first start with the Penicillium brevicompactum. So despite its name as part of the Penicillium genus, Penicillium brevicompactum is a type of fungi, first originated from a fungus culture from Japan, but now is commonly known as the foliage properties in fruits and vegetables such as potatoes and apples. The fermentation of Penicillium brevicompactum causes the production of secondary metabolite mycophenolic acid that is known to have an immunosuppressive activities and antibiotic activities against certain bacteria. Now, in its natural state, mycophenolic acid is active but toxic and therefore must undergo esterification process to make the compound safe to consume. If we have a look at the chemical structure of mycophenolic acid, it possesses a carboxylic tail at the very end, and as it undergoes esterification, this carboxyl tail will be transformed into an ester side chain, and therefore the compound is now given a new name, mycophenolate mofetyl. Now, mycophenolic acid is also available in its sodium salt form, in which the original chemical structure of mycophenolic acid is now attached to an additional sodium atom, which forms the sodium salt and is available in the form of white crystalline powder that has a high solubility at physiological pH and is widely used to prevent tissue rejection following organ transplants, as well as for the treatment of certain autoimmune diseases. Now, mycophenolic acid has also been seen to show inhibition of the gram-positive bacteria Bacillus anthracis, which causes anthrax, as well as the inhibition of osteosarcoma cell growth. which is the bone cancer that grows from within the cell. Therefore, mycophenolic acid is shown to be a potential osteosarcoma chemotherapy. Now, the fact that this mycophenolic acid metabolite is derived from fermentation process of fungi makes it especially necessary for it to undergo many additional biotechnological processes, such as purification and isolation from unwanted components. Now, the process itself has been summarized here in the diagram on the left, but I won't go to further details. However, I would mention, as I said before, that the mycophenolic acid is commonly known for its immunosuppressive activities. It is also shown to have the ability to prevent proliferation of T cells, lymphocytes, and antibodies formation, as well as the blocking and regulation of de novo biosynthesis of purine nucleotides. Here on the right, we have an example product that contains mycophenolic acid. This is called, this product is myfortic, and it contains mycophenolate mofetil. It is an immunosuppressant medication generally used by patients following organ transplantation, such as an allogenic kidney transplant. Now, the drug works by suppressing any potential rejection that may potentially cause organ failure. Okay, next I will talk about the Chlorella fulguris. It is actually a type of chlorophyll, which is an algae known for its green color due to the high chlorophyll compound it contains. Though often found in freshwater and wastewater, Fulguris is best known for its ability to undergo self-sporulation in autotrophic, heterotrophic, and its optimum medium, the mixotrophic environment, which results in the high-yield, high-rate metabolite productions. As an algae, Fulguris undergoes complex and various extraction pathways, some of which through centrifugation and pressurized liquid extraction. Now, C. fulguris is also popular for the primary active compounds it synthesizes, which includes proteins, lipids, and a range of vitamins, such as vitamin C, B12, and 6. Now, the secondary metabolites it synthesizes include a range of pigments, flavonols, and catechins, which are some of flavonoids with extensive health benefits that are necessary in our diet. And last and most importantly, the secondary metabolite beta-1,3-glucan. In its natural state, it exhibits immunostimulatory activities, but it is insoluble and thus must undergo carboxymethylation and form a carboxymethylglucan, its water-soluble polysaccharide. The increase in solubility, in addition to its antioxidant activities, allows for it to be used in the cosmeceutical industry, mainly for the topical formulation. As a nutraceutical product, beta-glucan is also a powerful component used to lower blood cholesterol levels by lowering cholesterol absorption rate.
Okay, so let's talk about lovastatin. What is lovastatin and what is the use of lovastatin in pharmacy? So lovastatin is a drug that decreases cholesterol levels in blood. Lovastatin is considered as a eukaryotic fungal, and then it is formed from two kinds of fungi, which is the Aspergillus terreus and also the Monescus rubber. So the commercial production of lovastatin is based on the submerged fermentation, but nowadays the solid fermentation, a solid state fermentation is more used in today's life. So the lovastatin is also considered as a prodrug lactones. It is hydrolyzed in the gastrointestinal tract into an active B-hydroxyl derivative. It is also a secondary metabolite produced from the ketide. So in pharmacy, lovastatin is mainly used for anti-hypercholesterolemia. It is used to decrease the cholesterol levels in blood. But there are other potential use of lovastatin. It includes therapy of Alzheimer's disease, cancer, osteoporosis, and so on. And then it also inhibits HMG-CoA reductant. Okay, for this, let's talk about on the next slide. Let's talk about what does the inhibition of HMG-CoA reductant mean? So HMG-CoA is also known as B-hydroxy B-methylglutaryl-CoA. This process occurs in the liver. The aim of this process is to synthesize the cholesterol. HMG-CoA or HMG-CoA first is produced from the condensation of the acetyl-CoA and the acetoacetyl-CoA. When the HMG-CoA forms, it needs to form into a mevalonic acid. In order for the HMG-CoA to turn into mevalonic acid, it needs a special enzyme. The enzyme is called the HMG-CoA reductant. An HMG-CoA reductant is also known as a lovastatin. A lovastatin is needed to inhibit the enzyme from turning into mevalonic acid. Why should we inhibit the enzyme? When a mevalonic acid is formed, it will start to make a mevalate path, and that path is going to produce more cholesterol. While the aim of our process here is to synthesize the cholesterol, not to produce the other cholesterol. So not only lovastatin can do this job. Lovastatin is the group of the statin drug. There are other statin drugs such as the fluvastatin, pravastatin, rosuvastatin, and stimulvastatin. Okay, so the brief explanation about chlorella vulgaris has already been explained in the previous slide. Now, I'm going to talk about the chlorella vulgaris uses in pharmacy. The carboxymethyl or CM is used in the cosmeceutical as for its role in stopping antioxidant depletion in the skin cells upon UV radiation, and therefore it shows anti-aging properties. Furthermore, its ability to promote the keratinocytes growth, skin epidermis renewal, and promote smoother skin made it a suitable active compound for topical cosmeceutical formulations. The C. vulgaris, minerals, vitamins, and chlorophylls is also marketed as a dietary supplement. As for example, this picture is an example of the chlorophyll-based dietary supplement.
This is another example. Aspergillus nidulans that produces a secondary metabolite echinocardin B. First, it is isolated from soil, plant debris, water, and air, and then it will be exposed to a chemical called SCIB. Then the secondary metabolite echinocardin B will be produced. Then this secondary metabolite can be used as an antifungal drug, which inhibits the beta-1,3-D-glucan synthesis in fungal cell wall. How it's extracted. A one-step microporous resin adsorption chromatography is involved in the extraction of ECB from Aspergillus nidulans fermented broth. And in pharmacy, it is a type of lipopeptide antibiotic. It is an antifungal drug which contains a lanol side chain that inhibits an enzyme that synthesis beta-1,3-D-glucan synthase required in the biosynthesis of fungal cell wall. ECB can also be modified by enzymatic dealkylation to remove linoleal side chain from the structure and perform sequential chemical reacylation to generate a few therapeutic antifungal agents for clinical practice, such as anidulafungin. Another role is that it is a host for recombinant proteins. Recombinant protein is a type of modified protein encoded by a recombinant DNA or modified gene. Low eukaryotic cells are chosen as a host for production of recombinant proteins because of several reasons. Because of its high growth rate, genes can easily be manipulated, and it has the correct protein processing and post-translation modifications. Now I'll give an example of a low eukaryotic cell used as a host for recombinant proteins. Pichia pastoris glycoprotein. First, its cellular pathway is re-engineered to mimic the N-glycosylation to produce the glycoproteins. Then, a combinatorial genetic libraries were constructed to localize enzymes used to produce these glycoproteins. And the Golgi body is then fused with enzymes such as UDP glucose 4 epimerase and beta 1,4 galactosyl transferase to accommodate the activity in vivo. Then the N-glycoproteins are produced at high yields. But why do we use Pichia pastoris? It is an important host for heterologous protein production and it has a high protein productivity. In pharmacy, glycoproteins are involved in immunity. Specific glycoproteins can be used to determine blood type, and it also serves as a natural neutralizing target for antibodies production by the body in fighting off an infection and conferring some degree of future immunity.
In conclusion to our presentation and also our research, the secondary metabolites produced by lower eukaryotic cells plays an important role in the synthesis and discovery of drugs, because mainly the discovery of drugs is secondary metabolites. And also, the lower eukaryotic cells are host to many biopharmaceutical metabolites. This is the end of our presentation. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.